Let me quickly check if I can share the screen. I guess it's working, right?
Yeah.
Ao vivo.
Yeah. Okay. Yeah.
Good. So it's my pleasure to host the last SBI.ImunoTalks of this year. So there was an initiative of the SBI to bring together good science, excellent scientists, and we had excellent talks over the second semester of this year. So we are going to definitely keep this seminar series going on in the future. And it is a big pleasure for me to host the last seminar of this year, which is going to be delivered by Petr Broz. So Petr Broz is a friend of us. He has been to SBI a few years ago, and unfortunately this year we could not manage to have the meeting because of the COVID situation. But Petr was kind enough to accept our invitation to deliver a talk about the recent research he's doing. He's doing a fantastic work in understanding the molecular mechanisms of cell death, mainly like focusing on inflammasomes and Gasdermin. And I'm sure it's going to be a great talk. So Petr is a PI at the University of Lausanne in Switzerland, and he had a fantastic career. I mean, he was first linked to the University of Bern, and then he finished the PhD and moved for a postdoc in Stanford University in California. Did and cell death and inflammasome activation, important contributions at that time with Denise Monac, so it was a nice collaboration. Then he moved back to Europe and started his lab in University of Basel. There was a few years, he was a few years in University of Basel from 2013 to 2017. And then he got a position, accepted an invitation to move to University of Lausanne, which I think was a great decision, a smart decision from Petr, because it's really the place where the inflammasome were discovered with Jörg Chop and Fabio Martinon, which is still in University of Lausanne. So I'm sure it's a great place to work with inflammasomes and cell death. So with that, I'm gonna thank you again, Petr, for accepting our invitation. It's a big pleasure to host you here, and it's all your words now.
Okay. Thanks a lot, Dario, for the nice introduction, and it's a pleasure to present some of our latest results. So I'm a microbiologist by training, so I come with an angle from the microbes to the inflammasome. So I'll share the screen. Okay. All right. So does that work? Can you see the PowerPoint slides?
Yes, we do.
Okay. So I'll go into the presentation mode, and maybe I changed it. Okay. So I quickly changed to the pointer here. Here. Okay. Great. So today I wanted to take the opportunity to talk a little bit about the non-canonical inflammasome signaling and this pathway which we think we have now. Sorry.
We are seeing not the presentation mode of your talk.
Okay. So what do you see? Let me change. So what do you see now? You don't see the slides.
Oh yeah, now it's good. So you just have to go in the presentation, presenter view. Perfect. Yeah.
Maybe I need to go like this. What do you see? Slides?
We see a smaller slide and a few
okay,
slides.
That's weird. That's very weird. Okay. Let me unplug my second screen. Maybe that's causing the problem. Better?
Yeah, now it's good.
Okay, good. So then there was the second screen doing this problem. Okay. Good. Okay, so I want to talk about some new insights into the non-canonical inflammasome signaling. So let me quickly, bring everybody on the same page here. So the topic we are working on is innate immunity, and especially pathogen recognition. You all know that innate immune cells like macrophages, dendritic cells use pattern recognition receptors to recognize or to detect the presence of pathogens. And these can be either membrane associated, like here, or monitoring inside of endosomes or phagolysosomes. But they can also be found directly in the cytosol. And they detect, in most cases, pathogen-associated molecular patterns, so unique microbial substances that allow a distinction between self and non-self. And upon engagement or binding of these PAMs, they will induce, in most cases, a transcriptional response, where, for example, do NF-kappa B signaling to induce cytokine expression and inflammation. Now, what my lab is focusing on are cytosolic receptors and a special subgroup of these receptors that will induce also inflammation, but by a different mechanism. So in this case, we get the engagement and activation of cellular proteases, so-called caspases, that will drive the inflammation. And very typical, these caspases will also induce cell death of the infected cell. So we call this pathway the inflammasome pathway. We roughly distinguish between a canonical and a non-canonical inflammasome pathway. So the canonical pathway is better understood. So here we have receptors that are in the cytosol that will recognize the pathogen-derived patterns, or sometimes also endogenous danger signals. And these assemble a complex, which you see here, which is basically the inflammasome that we are talking about. And this complex is an activation platform eventually for the protease caspase 1, which is most famous in its discovery to be cleaving cytokines like IL-1 beta and IL-18, which you can see here, and promoting their secretion through an unconventional secretion mechanism, which we now know is linked actually to the second activity of caspase 1, which is to cleave this protein here, Gasdermin D. So this protein will eventually drive the cell death and the cell lysis by forming pores in the membrane that will promote cytokine release and also drive inflammation by killing the cell. Now, today I'll mostly talk about the second arm of inflammasome signaling, which is the non-canonical pathway, which activates a different caspase. So here we have caspase 4 activation in human cells and caspase 11 activation in mouse cells. And the major driver here is LPS, or lipopolysaccharide, that will activate this caspase. Now, this is a very busy slide here. This gives you a little bit an overview on how these canonical inflammasome pathways are activated. So they are much better studied than the non-canonical pathways. So for example, we know that also from work from Dario, that the NLRP4 and NAPES inflammasome participate in the recognition of flagellin and structural component of type 3 secretion subunits. So here, there is a direct binding between the NAPES and these substances here to drive NLRP4 activation. We also have a direct binding, for example, here for AIM2, which is a DNA sensor in the cytosol. And then there is more complicated ways of activating inflammasomes. So, recently, for example, the NLRP1B has been characterized, where NLRP1 serves as a pseudo substrate for example, anthrax lethal factor, which is a protease that cleaves it. And you have a partial degradation then in the proteasome, and one factor being released that drives inflammasome assembly. So this can be, so there is no direct sensing of any bacterial factor here, but what is detected is actually the activity of a bacterial protease or a bacterial effector protein. And a little bit similarly, it's the case for pyrin, which detects inactivation of the RhoA GTPase. And here, NLRP3 is probably the clinically most important inflammasome. Here, the activation mechanism is not yet fully understood, but it seems to respond to, for example, pore formation in the membrane. So overall, you see, this pathway seems to be quite well understood right now. So I also wanted to quickly talk about the outcome of inflammasome activation. So I told you that Gasdermin is a substrate of caspase 1, but also caspase 4 and 11, and this causes this cell death. So what is this Gasdermin protein? It is a protein that was first described in the early 2000s, but its function only emerged around 5 years ago, when it was shown that that Gasdermin D, one of the family members, is a driver of inflammasome-induced pyroptotic cell death. So at the moment, we have 6 members in humans, 11 in mice, which you can see here. And all of these proteins work a little bit or look very similar. So you have this N-terminal domain, which is a cytotoxic domain, a linker, and the C-term domain, which acts as a repressor domain on the N-terminal domain. So you know, as a full-length protein, this protein is inactive. Now, in 2015, the Gasdermin D protein was linked to pyroptosis, and the groups of Vishwa Dixit and Feng Xiao, both at the same time, in 2 landmark papers, showed that the Gasdermin D is cleaved by the caspase to induce pyroptotic cell death. And here, just to illustrate a little bit what is it when I talk about pyroptosis. So apoptosis is a programmed cell death that is thought to be immunologically silent, and you get the formation of these apoptotic bodies that you can see here. However, pyroptosis looks morphologically very different. So you also start with ballooning and blebbing of the membrane, but eventually the cell just enlarges and loses membrane integrity, and all the cytosolic content will leak out and drive inflammation. So it's a very pro-inflammatory cell death. And so if you see this type, if you have your cell culture, you start seeing this ballooning, okay, that's a very good sign that you have some kind of necrotic cell death going on, maybe pyroptosis as well. So quickly on the activation of the Gasdermin. So all the Gasdermins have this cytotoxic domain, which needs to be somehow separated from the C-terminus. And the first one to be understood is Gasdermin D, which we know is cleaved by caspase 1, 4, and 11. Here in this linker domain, and this separates the N-terminus and allows the N-terminus to cause pyroptosis. Recent work has also shown that caspase 8, work by us and others, have shown that caspase 8 can do the same thing under certain condition. And also caspase 3, another apoptotic caspase, can cleave Gasdermin. And most recently now, the granzymes has also been linked to the activating different Gasdermins. So granzyme B, which is used by killer cells to kill, for example, viral infected cells. These granzymes, when they are delivered, they can directly activate the Gasdermins to kill cells as well. So granzyme A and B have been linked to Gasdermins by now. So what does this N-terminus do? We can see here some atomic force microscopies of membranes targeted by the N-terminus of Gasdermin D. You can see that we have this pore formation in the membrane, and work from Hao Wu labs has shown that the structure here of the Gasdermin H3 pore. So we get the formation of a large beta barrel pore in the membrane that will basically kill the infected cells. So this is the cause of this pyroptotic cell death. So I quickly hint on that also. So why are inflammasomes important? So we mostly study them here in control of inflammation or in pathogen infections. They cause a controlled inflammation, but chronic inflammation has also been associated with inflammasomes. So too much pyroptosis, too much alpha beta has been linked to many human diseases, from cancer, autoimmune diseases, neurodegeneration, sepsis, and so forth. And it is this NLRP3 inflammasome that is important. But for example, for sepsis, it's mainly the non-canonical inflammasome pathway, which I'll focus on today. So, I hope this gave you a little bit an overview on the field, on inflammasomes, and where we are working on. But today I want to focus on one story that we published recently, earlier this year, which is really dedicated to the non-canonical inflammasome pathway. And this story was mainly worked on by 2 postdocs in the lab, José Santos, who's a cellular microbiologist, and Dave Boucher, which is a biochemist. So the story starts a long time ago. It was already in my postdoctoral work, where we started to work on LPS recognition by caspase 11. So let me quickly reintroduce the LPS detection in general. So you certainly know that TLR4 is the main receptor for LPS that is found on our cells, and TLR4 will detect extracellular LPS, coming from gram-negative bacteria, for example. But there's also work by many groups have shown that TLR4 does not work by itself in this recognition pathway. So recognition by TLR4 requires other proteins as well, like LPS binding protein here, which will shuffle LPS molecules to CD14, and CD14 will then move them over to the MD2 TLR4 complex to trigger signaling by MyD88 and TRIF. So you can see that this requires several proteins, and the reason for this is that LPS is very hydrophobic. So there's no free LPS moving around, so you need to bind it and you need to bring it to the receptor. Now, when we compare intracellular LPS sensing by the non-canonical inflammasome to this, we know much less. So we know that caspase-4 is activated in human cells, or caspase-11 in mouse cells by LPS, which cleaves Gasdermin and will kill the infected cell. But we don't know if there's other proteins here involved in this pathway, like something similar like LBP that will shuffle LPS and bring it from the bacteria to the caspase, or if this works somehow differently. Work from Feng Xiao's group has shown that there's actually a direct binding of LPS here to the CAR domain of the caspases, which is necessary for the activation of the caspases. Now, why would there be other proteins? We also know that caspase-4 and 11 actually recognize the lipid A moiety here, which is embedded in the bacterial membrane and is poorly accessible to the caspase. So this raises, of course, many questions. So how does caspase-4 gain access to this part here? Any cofactors, or is there a platform for caspase-4 or 11 activation, sort of in analogy to the canonical inflammasome, some kind of complex that is built? So these are all questions that is at this point was relatively unclear. Now, so what we knew for some time, however, is that interferon signaling seems to be essential for non-canonical inflammasome activation. And this was some work I've started during my postdoc at Stanford. So, for example, here you can see mouse BMDMs infected with Salmonella. Wild-type macrophages, they die, release LDH, so they induce pyroptosis, and the caspase-11 knockouts don't die. But we also see that the IFNAR knockouts and STAT1 knockouts, so they lack the type 1 interferon pathway in this case, will not be undergoing this cell death as well. And this is caspase-11 driven death. So it suggests that caspase-11 activation in the mouse required type 1 interferon signaling. So this, over the years, led us to identify a group of protein that we think are absolutely essential for caspase-11 activation. And these are the so-called guanylate binding proteins, or GBPs, which you can see here. So the GBPs are encoded on two loci in the mouse, on chromosome 3 and 5. There's 11 of these GBPs here. And in the humans, you have 7 of them all in the same locus. So what are these proteins? So these are large GTPases around 65 kilodalton, which have a large G domain or GTPase domain, and this stalk-like region showed here. Some of them have a membrane anchor. And these proteins are very highly induced by both type 1 and type 2 interferons. They are related to the dynamins, so they are thought to somehow be binding to membranes and modulate membranes, but it's not so clear exactly what they do. But what has been shown over and over again that they are extremely important for host defense in vivo, for example, against bacteria and parasites. And they work in a cell autonomous manner, so they somehow block pathogens from replicating within cells. So work that I won't go in too much detail in the past has, for example, allowed us to show that GBPs are important for inflammasome activation during Salmonella infection here, or infection with Citrobacter, which both rely on caspase-11, at least partially here. So we use this chromosome 3 deficient mice, so they don't lack the GBPs on chromosome 3, and you can see the reduction here in cell death induction and also in caspase activation here and here. And also in vivo, GBP chromosome 3 deficient or even GBP2 deficient mice are highly susceptible to infections, for example, with Francisella novicida. And you can see that here, so caspase deficient mice have higher CFU counts than the wild type. They also die from the infection while the wild type survives. And similarly, the chromosome 3 knockouts, they die from the infection while the wild type survive, and they have higher CFU counts. So overall, this established the importance of the mouse GBPs for activation of the non-canonical pathway, so mostly caspase 11 in this case. But at this point, we didn't have much idea like how important actually are the human GBPs for activation of caspase 4 in the human system. So we asked if GBPs also control the non-canonical inflammasome in human cells. And so Jose, he started out with infection using our favorite bacterium, which is Salmonella. So we just use it as a perfect model for a gram-negative bacteria infection. So Salmonella can invade a number of cells, macrophages, epithelial cells, and it first ends up in a vacuolar compartment, but soon it also escapes from this vacuolar compartment and can also replicate directly in the cytosol. So Jose took a number of different cells like HeLa cells, but also HBECs, THP1s, U937, so monocytes or epithelial cells, and tested if there is any, primed them with interferon or kept them naive before infecting them with Salmonella, and looked at cell death induction. And what he could observe is that after Salmonella infection in wild-type cells, he could observe induction of cell death, which was dependent on prior interferon gamma priming. So you can see that here, the gray bars, interferon gamma prime versus the naive cells here with the white bars. And he also checked if this is activation of the non-canonical pathway. So we know that Salmonella induces the non-canonical pathway in mice or in mouse macrophages, for example. But at this point, we didn't know that for human cells. So he CRISPR'd out caspase 4 and Gasdermin D. You can see that we lose this interferon-dependent cell death here. So we next asked, okay, is this GBP1 dependent, this activation of this caspase 4 dependent cell death? So we did a CRISPR of GBP1, which we expected to be the most important of the human GBPs based on some other work. And indeed, the interferon gamma dependent cell death that we see after Salmonella infection was gone as well. But is this, at this point we asked, okay, so is this specific for infection with bacteria, the requirement for GBP1, or is this a general requirement for the non-canonical inflammasome pathway? So we went to transfect LPS into cells. So when we transfect LPS, we just deliver LPS in form of, you complex it with a transfection reagent, so we deliver it in the cytosol. And to our surprise, actually, also the cell death induction by the LPS transfection was interferon gamma dependent and dependent on GBP1. So this suggested that GBP1 controlled caspase 4 activation. And indeed, this can be confirmed. So we can blot for caspase 4 processing. So this are difficult blots to do. Sorry, difficult blots to do. But you can see that in wild-type cells, let's do it like this. So in wild-type cells, we get this p32 band of cleaved caspase 4 only when we transfect LPS in interferon gamma prime cells and not in unprimed cells here. And this is gone in the GBP1 knockout. Okay, so this established that the human GBPs were also important for activation of the caspase 4 pathway, so of the LPS sensing pathway in human cells. But how do GBPs work eventually? So, from the work that we have done in the mouse and other groups have in the mouse, over the years, a model emerged, which was proposed here in this cell paper by Kanaganti's group. And what you can see here is basically that they proposed that the GBPs work together with another GTPase, which is called IRGB10, shown here. So it was proposed that the GBPs together with IRGB10 target bacteria and eventually induce the lysis of these bacteria to release LPS, and DNA for inflammasome activation. Now, this model might be true for mouse cells, but there is one issue here, and this is that in IgB10 is not found actually in human cells. So, sorry, I am a little bit sick. It's very cold in Switzerland. So while we have the GBP family of GTPases both in the mouse and in human cells, the IgG family is only found in the mouse. So there's no IgB10 protein in humans at all. So how would the GBPs work in this case? So we decided to step a little bit back and do some more analysis on the human GBP family by doing first some stainings in Salmonella infected cells. So we use HeLa cells that express GBPs tagged with GFP, either in naive or interferon gamma prime condition to infect with Salmonella and do some fluorescence microscopy to see what happens. So I'll just show you first the interferon gamma prime cells. What you can see very easily is that the bacteria, which are here in red, are targeted by the GFP labeled GBPs. So GBP 1, 2, 3, 4 target really well. We can quantify that, while there's little targeting for GBP 5, 6, and 7 to the bacteria. So there seems to be this coat assembled around these bacteria that you can see very nicely here. Now, interestingly, this is in naive cells. All of them can target, but in prime cells, both all can target, but in naive cells, only GBP1 targets bacteria, which you can see here in the quantification. So this suggested to us that GBP 2, 3, and 4 required some additional factor that will help them to be recruited to the bacteria, and this factor was interferon inducible. And since GBP 1 didn't need anything else, we suspected that GBP 1 is actually the driver of their recruitment. And indeed, this was the case. So when we took GBP 1 deficient cells and compared them to wild-type cells, we could not see any recruitment of GBP 2, 3, or 4 to the bacteria, even if we primed the cells with interferon. And also, the inverse experiment here, when we take naive cells expressing mCherry GBP1 and co-express the other GBPs, GBP1 can drive the recruitment of GBP 2, 3, and 4 directly to the bacteria. So we have a situation a little bit like that. So we have Salmonella that are first detected by GBP1, which then gets the other GBP family members 2, 3, and 4 onto the surface of the bacteria to build this coat around the bacteria. So Salmonella has a little bit, I said, like is invading cells and goes through a vacuolar compartment and then later on escapes into the cytosol. So what is targeted by the GBPs actually? Is it the vacuole or is it the bacteria? So, and you can see that here. So there's the vacuole and here they escape. So they lyse the vacuole and they escape into the cytosol. Now to look into that, we use galectin-3, which will bind to the beta-galactosides on the inner leaflet of the vacuole, and it allows a very nice marking of the lysing vacuole during a bacterial infection. So we did co-stainings here, or used the GFP-labeled galectin-3 and mCherry-labeled GBPs to see if we have a co-staining on the same bacteria. And indeed, this was the case. So whenever we have a galectin-positive bacterium here or here or here, these bacteria also become GBP positive. And this is the case for all the GBPs as well. Now, important here is that actually there is no co-localization of the staining. So you can see it very nicely here. It seems that one part of the bacterium gets red and the other part gets green. So one part is galectin positive, the other part is GBP positive. And we interpret this as the following situation where there is a lysed vacuole protecting this part of the bacterium and being labeled by galectin in green, while the part of the bacteria that gets into the cytosol is labeled by the GBPs. So the GBPs we think label actually directly the surface of the bacteria. So mouse work has shown that the GBP labeled bacteria get somehow lysed. So we ask, okay, is this the case indeed also for the human, in the human system? So the human GBPs lyse bacteria when they get recruited to those bacteria. So we did this assay here in Gasdermin deficient cells. So these cells wouldn't undergo pyroptosis, because we know that the GBPs that the cell death induction was GBP dependent. So we used Gasdermin knockout cells to have a longer time to be able to monitor what happens after GBP recruitment to the bacteria. Do the bacteria get killed? And we did some time-lapse microscopy. So what you can see here are bacteria in red, so Salmonella in red in cells, GFP, GBP1 in green, and the composite of this. So when I run the movie, what we will see is that the bacteria are targeted very nicely by the GBPs, but they just continue replicating. They lose a little bit the cherry signal, but they seem to be still fine and can replicate. So it doesn't seem that the GBP recruitment really killed off the bacteria or lysed the bacteria in this way. So if the GBPs if human GBPs do not work like it was proposed for the mouse GBPs by lysing a targeted bacterium, so how do they actually work? So how do they promote LPS-induced caspase-4 activation? So the answer actually to this question came when we started to look at the subcellular localization of caspase-4. And actually, to our surprise, very few people have done that for the non-canonical pathway. So everybody has done that for caspase-1, and you can see caspase-1 being recruited to ASE specs, to inflammasomes, but for caspase-4, that was not done. So we used caspase-4 GFP, shown here, expressing HeLa cells, and infected those cells with Salmonella, interferon primed or naive, and did some microscopy as usual. And what we detected is the following. So in interferon gamma primed cells, we saw that caspase-4 also assembled a shell around the bacteria. So the caspase got recruited to the bacteria in those cells. But this could not be seen in naive cells, so not interferon gamma primed cells. And indeed, this can also be quantified. So usually around 10 to 20% of Salmonella get caspase-4 positive. And it's not visible in the naive cells, only in the interferon gamma primed cells. So also we did the same galectin staining experiment to see is it the bacterium itself or is it the vacuole of the bacteria that is stained. So again, we use galectin here in this time galectin in red and caspase-4 in green. And did this co-labeling. You can see it here, the bacteria get labeled by both. And we get also this partial staining here for the lysed vacuole in galectin-3 and the surface of the bacterium being labeled by caspase-4 here in super resolution microscopy. So we have a situation like this where we have vacuole lysing and the surface of the bacteria as, attracting caspase-4 in some way. So of course, at this point, things started to make sense. So, putting GBPs were recruited to the surface of the bacteria, the caspase as well, and the GBPs were necessary for caspase activation. So we asked if the GBPs are necessary for caspase recruitment to the surface. And indeed, this was the case. So whenever we have a GBP positive bacterium here in red, this bacterium will eventually also become caspase-4 positive, shown here in green. So it takes some time. So there's around a 5-minute lag. So here at 48 minutes post-infection, we get GBP, or post-start of imaging, we get GBP recruitment. And then 5 minutes later, we start to get the caspase-4 recruitment. And eventually the cell dies. So you can see here the ballooning of the cell membrane. This can also be quantified. So in the GBP, in the wild type, we get around 20% of caspase-positive bacteria upon priming, and this is completely gone in GBP1 deficient cells. So if you don't have the GBPs, there's no caspase recruitment. So we have a situation like this where Salmonella is first recognized by GBP1, which recruits them 2, 3, and 4. And this complex then will recruit caspase-4 to the surface of the bacterium as well. So which proteins in this complex are necessary to recruit the caspase? So is it just GBP1 or does it need 3 and 4 as well to do it? So we looked at that by using HeLa cells in this case, where we co-express mCherry GBP1 and caspase-4, and either we are in the naive conditions where no other GBPs are made, or in the interferon gamma prime condition where other GBPs are made as well. And what we could observe is that while GBP1 gets recruited to the bacteria in both cases, both in naive and interferon gamma prime cells, the caspase is only recruited in interferon gamma prime cells. So GBP1 by itself cannot recruit the caspase. It needs the action of the other interferon-induced GBPs. So which are these other GBPs out of these, out of 2, 3, and 4 that are required for caspase recruitment? So I want to go into details here. So we tried, many combinations of expressing different proteins to reconstitute the whole system in naive cells. So what is the asking basically, what is the minimum requirement of GBPs to get caspase-4 recruitment to the surface of the bacteria in naive cells, and to get LDH induction, to get caspase-4 dependent death? And to make a long story short, basically what we see is that if we that GBP1 here together with expressed together with 3, or GBP1 expressed together with 4, is sufficient to get the recruitment of caspase-4 to bacteria. So you can see that here, and this never reaches really the level that we get in interferon gamma prime cell, but it's quite good. And the activation is a little bit similar. So when we look at cell death induction, it's basically GBP1 together with GBP3 and 4 that drive the induction of cell death. So GBP1 with GBP1 with 3 and 4 gives cell death in naive cells. So this means that the minimal components we need to activate caspase-4 in the human system is, of course, the bacterium and the LPS. We need GBP1, and we need 2 other GBPs, GBP3 and 4, to drive recruitment and cell death induction. So these are the minimal components of the pathways. What GBP2 is doing in the whole system, we don't know, but it doesn't seem to be really important at all. But it's recruited to the bacteria. So to summarize this part, so we have the bacteria escaping from the cytosol. They get targeted by the GBPs, which build this platform or a signaling complex on the surface of the bacteria, and the signaling complex recruits the caspase and will somehow drive the activation of the caspase. So we published this data, and in parallel another group published similar data at the same time as well, confirming basically our results that the platform is assembled by GBP1, 2, 3, and 4 and activates caspase-4. But of course, this raises a lot of questions. So now, like, what does this GBP complex on the surface of the bacteria actually do? Does it transfer LPS somehow from the surface of the bacterium to the caspase to activate the caspase? Does it rupture the membranes to liberate LPS molecules? Is there a direct interaction between the GBP complex and the caspase at all, or does it destabilize the membrane and the caspase can come in? And actually, what is it actually that the GBPs recognize themselves? So we know that the caspase can bind LPS, but what do the GBPs, or GBP1 actually, which is the first GBP to go, recognize? So in the last 5 minutes, I quickly want to answer this question here. So what does GBP1 recognize? So while doing all this work, we, of course, not only looked at Salmonella, and we looked at many different bacteria and their recruitment or the targeting by GBP, some gram-negative bacteria, and also some gram-positive bacteria like Listeria here, but gram-negatives like Shigella, Burkholderia, Salmonella, Francisella, and so forth. And, for example, Burkholderia is beautifully targeted as also by GBP1, 2, 3, and 4. And the same is the case for Shigella. But for gram-positive bacteria, so for Listeria, we never see any GBP targeting. So this started, a pattern seemed to emerge here that, gram-negative bacteria have something that allows GBP recruitment. So what is the defining feature of gram-negative bacteria is, of course, LPS, lipopolysaccharide. So we wondered if the GBPs actually interact with LPS. and started to do interaction experiments. So in this case, this is one pull-down experiment where we basically express GFP-tagged GBPs, 1, 2, 3, 4, or 5, or 4. We prime with interferon, we lyse those cells, and we spike in LPS biotin, or it was biotin as well, itself as a control, and we pull down the biotin, LPS or biotin with streptavidin magnetic beads, and we check what has been pulled down. What you can see is that among the 4 GBPs, we basically get a strong pull down of GBP1 by LPS biotin, not by biotin, a little bit GBP3 here as well, but not 2 and 4. So this serves as an internal control for this assay. So this suggests that GBP1 has the ability to interact with LPS. Of course, these pull-down experiments are dirty, so we went into purified proteins here. So we did surface plasmon resonance studies to really be able to verify this interaction between LPS and GBP1 with purified proteins, as it has been done, for example, for caspase 11 and caspase 4 and LPS by Feng Xiao's lab. And you can see these binding curves that you can get, and it allowed us also to determine the KD of this interaction, which is around 63 nanomolar. And this is indeed a little bit in the range what has been published by Feng Xiao for caspase 4 LPS and caspase 11 LPS interaction. So it's a similarly strong interaction with LPS. So we wanted to explore this further. So we set up a cheaper assay for this interaction because the surface plasmon resonance is quite expensive. So here we basically take recombinant monomeric LPS-free GBP1, and we either incubate it alone or we incubate it with LPS, and we run it on a Cephadix 200 gel filtration column. And this is what we get. So we get these peaks from elution from the column. So if you have just GBP1, we get peak here running as a monomer, so you can see that on the plot here as well. And if you mix with LPS, it moves to higher molecular weight fraction. So we get a smear here to higher molecular weight. And this is actually, the LPS micelle runs around here. So this is binding, what we get here is the binding of GBP1 to the LPS micelle. So this allows us to very quickly check what disturbs this binding or what is necessary for this binding, this type of assay. So what is necessary about what is recognized on LPS by GBP1? So you certainly know the structure of LPS. You can see it here. So we have this lipid A part here, hydrophobic, which is recognized by the caspase. A lot of negative charges here, inner and outer core sugars, and the variable O antigen depending on which bacterium you are in. So this is LPS from E. coli. So we speculated actually the binding could be mediated here by the negative charges, which you can find on LPS. And to look into that, we use different approaches. So for example, we incubated, so we did this gel filtration assay. So here you have GBP1 with LPS alone. So we get still a little bit monomeric protein, but here is oligomeric protein binding to LPS. So we were incubating the LPS with calcium ions, so which would hide the negative charges, and we go back to more monomer. We can also incubate with polymyxin B, which will disrupt this interaction by binding to LPS as well. So there's a competition between polymyxin B and GBP1 for LPS binding, and you can see that the orange curve compared to the black curve, we go back to the monomeric protein. If we dephosphorylate LPS, we actually go back to the monomeric protein as well. And even, what is important, dephosphorylated LPS is not able to engage the non-canonical pathway. So if you transfect dephosphorylated alkaline phosphatase-treated LPS, basically we don't have any activation of cell death. So it seems that these negative charges on LPS are important for this interaction. So what is important on the side of GBP1? So here we looked a little bit into other LPS binding pattern recognition receptors like LBP, for example. So LBP, I've shown you, shuffles LPS from bacteria to CD14, which then brings it to TLR4. And what is important in LBP is that it has positively charged surface residues, which can interact with this negatively charged LPS. And you can see that here, these are LPS micelles, these worms, and the smaller white worms here are basically this LBP molecule, and it binds through this positive charges to the LPS micelle, and then extracts single molecules of LPS. So we did some structure-based analysis of GBP1, so here GBP1, and identify surface patches that are also positively charged in GBP1. So A patch, B, so these are mostly surface-exposed positive residues. Mutated those and did the gel filtration assays to see which one disrupts interaction with LPS. And some of them don't have any influence at all on LPS binding, like this mutation here of the KLKK locus here. Some of them have a minor impact, but for example, the mutating this 3 lysine 61 to 63 to alanines disrupts the binding, and you have more monomeric protein again, which can't bind to LPS. And we also checked this mutation also in vivo. So for example, here are cells expressing this mutant protein compared to the wild type, and another mutation here. And what you can see is that we lose the binding to the bacteria if we mutate this patch here, a patch here on that part here of the GBPase domain. Okay, so with that, I quickly summarize. So what we think we have found is a new family of pattern recognition receptors, the GBPs. Actually, GBP1 of one of the defining members of this family. It is an LPS binding protein. It's probably the first protein or pattern recognition receptor that recognizes a gram-negative bacterium in the human cell. Sorry, it will decorate this cell here and recruit the other GBPs to build this platform that will then recruit a second receptor for LPS, which is caspase-4. Now, exactly what happens here at this point where the GBPs promote caspase-4 activation, we don't know, but the outcome is clear. The caspase will be activated, and the infected cell will be killed and cleared by pyroptosis. So, we are currently looking more into what is going on this level here. We have also shown that only GBP1 to 4 are involved in gram-negative bacterial recognition, but what are the other GBPs doing? We have GBP5, 6, 7 here in the human system as well. We don't know. Interestingly, also GBP1 not only recognizes bacteria, but it also recognizes parasites like Toxoplasma, for example. So what is it on the parasite surface that attracts GBP1 to the parasites? And also want to highlight that we are not the only one that had this year worked on this pathway. So it was us and Felix Rando's group, but also Jörn Kerr's lab basically has also shown this interaction between GBP1 and LPS this year. So it's good to be also confirmed by other researchers in the field. Okay, so with that, I'll quickly just highlight who, again, Jose and Dave, who has done the work. So you can see them, these guys here, our funding, and thanks a lot for your attention. I'm happy to answer questions. Okay.
Thank you very much, Peter. Okay. It was an excellent presentation, as expected. It's a very nice story. So I will start with a question from Karina Bortolucci. So Karina says, "Hi, Peter. I'm very happy to have you here. Congratulations on the beautiful work. I wonder if caspase-1 or caspase-8 could also be recruited to the bacteria. Hope to see you soon."
Well, I hope to be able to go to Brazil next year, maybe for a meeting again, and hope to see you guys soon. Yeah, so that's a good question. So we have ourselves not looked into that, but our friends, competitors, Felix Rando's lab did some experience, and there was no recruitment of caspase-1 to the surface of cytosolic bacteria, and also not caspase-8, if I recall. We see a little bit recruitment of caspase-5. So you know that in the human system we have 2 non-canonical inflammasome caspases 4 and 5, but it's mainly caspase-4 that is recruited. There's never so much 5 being recruited.
Great. So I have another question from Warison Andrade, the famous Little Milk. He says, "Great work. You have clear data showing that the role of GBP on the activation of caspase-4 in bacterial infection." Mm-hmm. So he asks, what about transfected LPS? Do you still need GBP1 2, 3, and 4, or just GBP1 is alone, it's sufficient to bind?
Yeah, no, so we see that, I showed a little bit the data as well. So LPS transfection is also fully GBP1 dependent, but we have, which was a little bit surprising to us, but we imagine that when you deliver LPS with a transfection reagent, it's actually, it is a liposome, right? So the LPS is still embedded in the membrane-like compartment, and to activate caspase-4, you need the GBPs. And even if we transfect LPS, for example, if you take FITC LPS and transfect it, you can see that this FITC LPS gets targeted by all the GBPs as well. So the GBPs treat the transfected LPS as a membrane. Now, it's a little bit different if you electroporate LPS. And this, basically you deliver it, you disrupt the LPS micelles at that point. And then the requirement for GBPs gets less strong, but it's still detectable. So really I think what the GBPs really are necessary for is actually to make this membrane integrated LPS accessible for the caspase. How they do that, I'm at this point not able to answer.
Great. I have another question here from Joseli Lanes, who is a immunologist from Rio de Janeiro, very active in the society as well. So in this system, present in different, if this system is present in different cell types, such as astrocytes and microglia?
Good question. So we have looked at around 10, 12 different lines of human cells and also primary macrophages. There, it's absolutely the same. So the system is present there and it's absolutely required for LPS recognition in the cytosol. We haven't looked in astrocytes or microglia, I have to say, but I expect that it will be working there similarly. So there is, I think I lost you. So no caspase-4 activation in human cells without GBPs and interferon priming in our hands.
Cool.
Yeah.
So I actually have a question as well. So it was very attractive, the hypothesis that GBP is somehow involved in lysing the vacuole. And this, I think there is really convincing data in the toxoplasma field showing that GBP somehow lyses the membrane itself. So in your view, Peter, how do you see this process? Do you think is there any protein from the host side that is involved in lysing the membrane if it's not GBP? So how do you see this process?
Well, that's a good question. So certainly the toxoplasma data are pretty clear that there is lysis of the vacuole and that the GBPs have a membranolytic activity somehow. And we, maybe they indeed need to disrupt somehow a little bit the outer membrane of bacteria to, expose the LPS. But I think it's something that is mainly observed in macrophages. So there seemed to be something else, I don't know, in the mouse system could be IgB10 that is required for this activity. In human cells, I don't know what it could be that would provide the GBPs with this membranolytic activity. But at least, in the epithelial cells that we looked at, for example, the HeLa cells and other cell types, there was the recruitment of the GBP was not to the vacuole, but really to the bacteria. So we will go back to the mouse system and to re-examine our own data from years ago, because at that point we saw GBP positive bacteria, but we never checked if it was the vacuole or the surface of the bacteria. So we need to go back and re-examine that to really make sure what's going on and really go again into IgB10. Is IgB10 really important in the mouse system or not, right? So this, so what would it attract to the toxoplasma vacuole is weird, so is unexplained to me. I think there must be some toxoplasma lipid, highly negatively charged toxoplasma lipid that is in the vacuole, that is also found on the surface of toxoplasma, that will attract the GBPs, because once the vacuole is lysed, the GBPs will attack the pathogen and destroy the pathogen membrane. Yeah.
Great. So I have a question from Priscilla Campos. She says, great talk. Can we speculate on the role of GBPs and caspase-4 in MTB infection, since the bacteria is like...
Yeah, so it's something in between gram-positive and gram-negative. Great question.
Exactly. This is what
So based on published data from the lab of John J.D. McMicking that was published almost 10 years ago, Mycobacterium bovis, I think, is targeted by GBPs. So that would suggest that, the GBPs have the ability to recognize something there. I don't know what it is. We haven't ourselves tested it. So, but I guess while they can't recognize, at least let's put it like this, GBP 1 to 4, which is the GBPs that we looked at mainly, don't recognize gram-positive bacteria. But maybe other GBPs recognize gram-positive bacteria and mycobacterium, sorry.
Yeah. Just linking to this question of Priscilla, I mean, you probably follow that paper saying that lipoteichoic acid from gram-positive bacteria triggers an NLRP6 to recruit caspase 11 to the vacuole. There's a cell paper from Gabriel Nunes. So did you ever test that if GBP 5, 6, or 7, they somehow may be involved in the precognition of gram-positive, since you have the with gram-positives as well?
Yeah, we haven't really seen also recruitment of neither 5, 6, or 7 like it's, but maybe this is something that could be conserved in the mouse system, because in the mice we have more additional GBPs. We have 11, while in the humans we have only 7. So we haven't looked at the mouse GBPs, if any of those might be able to recognize the positive bacteria. No, how this LTA would activate NLRP6 caspase 11 is of course a different question, and has not been looked at if this requires GBPs, but would be attractive to do. Yeah.
So that's the next question is Thiago Mineu. Hi Peter, great work. Have a clue if GBP have a common binding site to the ligands?
Can you repeat the question? You froze for a minute.
Do you guys have a clue if GBPs have a common binding site to the ligands?
So at this point, we know that there is a couple of positively charged surface patches on GBP1 that are required, both in the large GTPase domain and at this very C-terminal. So you have this CAX box where it's isoprenulated to allow an insertion in the membrane. There's also positively charged residues. And these seem to be required, at least for GBP1. And you don't find these in the other family members. So GBP2, yes, a little bit, but 3 and 4 doesn't have the same positively charged residues. So, and these are the ones that can't really interact with the ligand, but interact with GBP1. So they probably heterodimerize or oligomerize with GBP1. So this ligand binding activity is only found in certain GBPs. So I would say we have a hierarchy, right? So we have 2 layers of GBPs. The ones that go first, this is 1, 2, and 5, which are the ones that have a membrane anchor. And then the other one that come later, which is the second layer. And the third layer would be then somehow caspase activation. Like, receptors, ASC, caspase 1.
The same.
Somehow. Well, that's ideally.
Good, Peter. So the next question is from Gustavo Amarante Mendes. Hi, Peter. Excellent work. Do you know how far in the phylogeny goes the GBP as a pattern recognition receptors? And then he's finished with hugs.
Okay. That's a good question also. So they actually go back. Also, for example, a zebrafish is a great example. So you find them in fish, but then they become very interesting. So actually, then the structure of the GBPs changes. So in zebrafish, for example, you have GBPs that are directly fused to card domains, which is super surprising. And they also participate in inflammasome activation there, but maybe more directly in that they recruit the caspase. And so these ancient GBPs are a little bit different. So it looks like that from these ancient GBPs, which had the GBP function and the card function, these things got separated. So in our mammalian system, we have now the GBPs, and we have the caspases with the card. and they are sort of linked functionally, but how they are linked on a molecular level, we don't know, because the GBPs don't have a card anymore. So where did this card go? Is there a protein missing that connects these proteins? Yeah. So yeah, they go back. So mammalian cells are, vertebrates are not the only ones having GBPs. You can find them in fish, but also in lower animals as well.
Great.
They are different.
So, yeah. So open to the next question. So it's a little after 1 p.m. in Brazil and almost 5 in Switzerland. So we're gonna ask the last question. So it's from Rafael Correia. Amazing presentation. He would like to know if GBP could bind to the virus, such as COVID, Zika virus, and activates the non-canonical inflammasomes.
That's timely, given COVID-19. Actually, yes. So there is evidence that GBP5 in the human system is required for HIV, innate immune defense against HIV virus. There's quite a lot of paper which show that it blocks, that it binds. There is this replisome of the virus, sort of binds there and prevents its replication. How this works, like what it binds, how it does it, if it oligomerizes, is not known. But so there is a link between GBPs and viruses. But if that activates inflammasomes, I don't know, or is unknown, right? And COVID, nobody looked at yet. Maybe we should.
Definitely should, yeah. All right, Peter. So on behalf of SBI, I would like to thank you a lot for excellent presentation.
Thanks again.
Delivered the talk. And this is the last SBI ImunoTalks of the year. And I'm gonna thank again Rafaela Garcia and all the team of the SBI, the journalists of SBI for the excellent seminar series we had this year. So we're probably gonna launch again in February. Keep tuned in the SBI social media. That we're gonna announce the following up seminars for next year. So again, Peter, thanks so much. And
thanks for the invitation and for the great questions. Okay, bye everybody. Stay safe these difficult times, and maybe next year we'll meet in person again.
Should definitely come. All right.
Bye, Dario. Bye, everyone.
Bye-bye.